carries genetic material.
All right, so it carries genetic material.
Looks like a ladder.
You have likely been told before it looks like a ladder. Any kind of ladder in particular?
Like a swirly one.
Yeah, they tell you twisted ladder. Truthfully, biology teachers everywhere tend to oversimplify it by saying twisted ladder. I would say the same thing if you had to ask me to describe it quickly, knowing that I actually prefer to think of it like a spiral staircase. How's that different than a twisted ladder?
It has more depth.
More depth than straight up and down?
Spiral staircase is twisting like it's twisting, or no, the twisted ladder is twisting with a spiral staircase. The where you're twisting around is in the middle of the climbing. It's like to the left of it.
Okay. So there's some strange structure, except there's this really weird staircase in Santa Fe. You guys ever have the opportunity to go out to Santa Fe, New Mexico? They call it the miraculous staircase. It's very confusing when you look at it. Like at first I was like, a spiral staircase, that can't be confusing, right? Like, spiral staircase, whoop-de-doo. And then you gotta pay the nuns $2 to go see it. And I'm like, I'm gonna pay $2 to go see some stairs. But the miraculous staircase, they didn't have a way in this church to get up to their choir loft, right? There was no ladder. And it was such a small church that they couldn't put in like a staircase, a proper staircase. So one day a man showed up who said he could build the staircase. Of course, his name was Joseph. And in a matter of a short amount of time, made a staircase out of wood that engineers couldn't figure out. Like, even modern engineers look at it and can't figure out how he could hand engineer this miraculous staircase. And then before the nuns could pay him, Joseph disappeared. You guys don't look like you're buying it. Bunch of skeptics. Some faith you have.
I believe it.
Wait, so he just disappeared?
What a legend.
Yeah.
Just disappeared.
He had to make it by, so it had to have been done by hand because this was like a while ago. It is a really pretty thing. Turns out, no, that it's now owned by like a spa and hotel, so it's very strange.
Did they take it out of the church?
Wait, what's it
Just
called?
bought the whole building.
Marvelous staircase. Miraculous.
Marvelous. I mean, Joseph is marvelous, but it's probably more of a miracle.
Wait, is it St. Joseph?
Chapel?
That's
So
what
then-
they.
So
Yep.
then he disappeared.
Yeah. There's also
It's
Well,
pretty.
they have a cool choir wall.
They do have a cool choir. It's a very pretty chapel. It costs like a small bajillion dollars to get married there, but you know,
Worth it.
I don't know about that.
Why would you pay so much for that?
But when you say spiral staircase, that's what I think of. Yeah, right? Crazy, right?
Wait, so
Pretty.
they haven't figured it out yet?
The docent, when I paid to see it, said they hadn't, but I don't see. I don't know. I admit my skepticism in as much as I was like, it looks like stairs. It's pretty, but whatever. But when I say twisted staircase, that's kind of what I picture in my head, those stairs. I should have had that picture in here, sorry. Fail. I anticipated other conversations later and embedded those photos, not this one. How does it do this thing? This thing you purport that it does, that it carries this magical genetic material? Unicorns? Rainbows? Rainbows, most likely.
Like, they've chosen, like, different sequences of, like,
Those words are all pretty important for us. It is a code. Sometimes instead of code, I could say recipe. It is about a specific sequence in that code. So just like the word cat can only be spelled C-A-T, certain recipes can be made with only specific ingredients. So it has to be a very specific section of code. And the ingredients that we happen to use here are nitrogen bases. These are all pretty important words for us. How many of you are like, yes, nitrogen base? I'm good with that. Nucleotide. How many of you are like, this sounds like October? It sounds like chapter 2. Dun, dun, dun.
I've been thinking too much about when it's like
You know, like it spirals.
out of control.
How long did you take to think up of that?
I don't know, but I said something punny in the hallway yesterday, and somebody offered me fudge, and I was like, oh, thanks, it's really sweet of you. And then I died laughing because I thought it was hysterically funny, and they just looked at me like, you are the stupidest human being who has ever spoken to me. And then I was like, and then I kind of wanted to cry. It's very sad. What else do you know? What did you brain dump?
Well, I knew the first one. That's what I said. Like it's genetic material that makes up who you are.
All right, so somewhere in grade school, you guys are probably all told genetic material makes up who you are. You get it from mom and dad, heredity.
Yes.
Yeah? Okay.
It resides in the cell.
It resides in the cell. Not only does it reside in the cell, it resides in a very specific small region of the cell called the nucleus. Just to make sure that I'm very clear about this one, every single nucleus in your body contains all of your DNA. All of it in every nucleus. So you get lots of copies. You started with one copy, and then that copy divided. And then that copy divided, those 2 copies divided into 2 copies, and then those 2 copies divided into 2 copies. Well, actually, it was those 4 divided into 8. And you blastulid from there. It's marvelous.
If the DNA replicates, how does it get
All 6 billion, I mean, I haven't told you 6 billion yet. How does it get to all of it?
Yeah.
You started as one original cell, and that one original cell made an exact copy. So then 2 cells had all of the DNA, and then they made exact copies of themselves. So those 2 cells turned into 4 cells with exact copies of all of it. But the process of doing so is just DNA synthesis. That's mitosis. Like that, like how do we have that many cells with all of them? Because you started as one cell. It's the afternoon and the internet. I'm cursing it in my head at the moment and shaking my fist violently. Can you tell? Oh. We all agree this is kind of where we're starting. Anything more complicated? Anything you're like, hey, I know this and I need to prove it right this instance? No. Oh, I was hoping that was it. Yay!
There we go, Cam.
You and Messelson install, right?
It's 2 strands of nucleotides.
2 strands of nucleotides in our double helix. Oh, fiddlesticks. I ran out of room. It is, in fact, in a double helix. Helix is just a shape, right? A twisty shape.
The pattern makes up the gene.
Oh, who knew I could do that? If I'd only paid attention at some point. The pattern of those nucleotides makes up a gene. Okay, we can do things with this. We can go places, kids. A little bit of history. Have I ever shared how I feel about history? It's kind of a bummer. You got to know it, because if you don't, it'll repeat itself. But nobody really likes memorizing dates. Gregor Mendel is an old monk. We call him the father of genetics. He's the guy who proposed that something from a parent is passed on to the child. That was late 1800s. Curses, you blazes, internet-based machine. It wasn't then until much later than that we had a word for stuff. We are approaching the 1950s when scientists finally give stuff a name. DNA. But they haven't any idea what it's shaped like. And there are scientists working very carefully to determine what it's shaped like. 2 scientists to this day are credited for the discovery of the shape of DNA in 1953, James Watson and Francis Crick. They get all of the fame and glory, the Nobel Prize associated with it. Ah, yes, that's Watson and Crick. But you'll notice I don't have a picture of Watson and Crick here. Oh no, instead I have a picture of the X-ray crystallography that led us to discern the image. We inferred what the image was based on the measurements we took off of this photo. People who did the measuring, Watson and Crick. Who took the photo? The woman next to it, Rosalind Franklin. So scientists to this day are very, very careful to credit her, because this was 1952 and a Jewish woman was in a laboratory working on cutting-edge science. 1952. Couple years after World War II. Kind of a big deal, right? So they aren't going to be particularly kind to her. They took her work and did not credit her for it. And by took, I mean literally stole.
Wait, where did this happen?
England.
Alright, I was gonna well, I mean, wouldn't they be like extra Jews since all this stuff just happens to them?
Prejudices run deep. You guys hear 1950s and you're like, that's like eons ago. Listen, don't tell my parents that. They're born in the 50s. They're going to be a little offended. It's not actually that long ago. So this is relatively new science, so much so that it wasn't until 2001 that we embarked on and completed the Human Genome Project. The word genome is the scientific term for every single nucleotide in an organism. So every single nitrogen base. In 2001, they finally put them all in order for the human. But just because they were in order doesn't mean we had any clue what they did. So imagine me handing you a Russian dictionary. You may have every word in Russian in your hands, but if you don't know the Cyrillic alphabet, do you know any Russian? They do, in fact, have a different alphabet.
Wait, so does this mean that you have the words in Russian and you could just look for the matching characters? Or does it mean you have the equivalents of what they would be in our alphabet?
You have the Russian characters in the Russian order. What do you do with it? Stare at it? Ask a small Russian child to teach you what the order is?
Couldn't you just use Google Translate?
You could, and that's the best part. What we currently have is the human genome, right? Actually, we have the genome of quite a few different animals now. And in 2001, when we got them, we had no idea what they meant, so people started using code to determine what it meant. They tried to crack the code to determine what sections of it do. There are still sections of it we don't know what they do. So there are scientists who are currently working to write computer programs to recognize patterns, to find where patterns occur among the genome. So those of you who are taking data and design this summer, this is essentially how it's going to work. You're going to take that information and put it in Google Translate. Except it's not Google Translate. You have to write the program. You can do it. I know you can because we've planned out how you can. So you're the scientists doing this work. Now you're like, trust me, it's a cool, cool place to be, and I wish someone had given me that opportunity, but nobody did because nobody loved me as much as DF and I love you. That's an awful lot. You guys are a hard sell today. Holy smokes, artichokes.
You just really scared us.
You shouldn't be. Shouldn't be. I'm so smart. There's a reason why we're doing this chapter leading straight into data and design. It's like they pay me to do this. That's funny because they do.
Do it.
Won't.
You're right, I need to drink it.
Do it.
So, 2015, we still haven't the foggiest, but all of them do. How many of you are like, awesome, it gives me something to do when I grow up? Listen, those people make good money. Don't judge. You can fill this out without me. That's what you're going to do. And the polymer. Yay. Sequences in series are usually more than 1,000 to 2,000 nucleotides long. So that's like crazy long, right? Like hugely long. Dogma meaning belief, right? So it is the central belief of all biologists that DNA codes for RNA, and RNA then is used to make protein. When I say that this is from now until June, it's the process of how first DNA makes a copy of itself, and then how DNA is transcribed into RNA, and then how it's translated into protein. The stretch of DNA in question is a gene. Gross oversimplification to say one gene for one protein. Turns out one gene can actually make multiple different proteins depending how you edit it. But, you know, things we learn with experience. You saw something vaguely like this, but my drawing version of it last chapter, where we started with a stretch of DNA like that. We took our DNA and we wrapped it around proteins called histones, and then those histones folded up 8 of them to make a larger little ball or a bead called beads on a string. And then they fold up to make a flower. And then that folds up to make a nice rope. Then that nice rope continues to tighten until we make something that looks like that. It's laying on its side, forgive it, it's napping. What is it? It's a chromosome. Most people misunderstand how genes fit into this big picture. God bless you. So, each one of, that's one nucleotide. There's 2 nucleotides, 3, 4. You have 6 billion total in every cell. But they're paired. So how long is it if they occur in a pair? The whole chain itself is 3 billion. So you have 3 billion, a chain of 3 billion of these things that get all wrapped up where, let's switch to that lovely bright blue. Let's call that 1,000 nucleotides. That little stretch of DNA would be 1 gene. That section. And then we have like a little section in between that we don't really know what it does yet. Back when I was your age, scientists called that junk DNA because they didn't think it did anything. But it turns out a few years ago they discovered it does stuff, so we can't call it that anymore. Oops. And then you have another gene. Oops. And then you'll have a little space and you'll have another gene. That's how this works. So all of your DNA is just a whole bunch of genes all stacked next to each other. What they do? Meh, we don't know. Well, not all of them. Some of them we do. But that's how it fits together for our group picture. We're going to break it down to the smallest part of that great big picture. The truly teeny tiniest part of that great big picture is the monomer. Have you seen this before?
Yes.
What is this? Oops. This part. Phosphate. And this part. So it's a sugar. Since we're looking at DNA, you are correct. It is called deoxyribose. That's what that specific sugar is. And of course, my darling, stunning honors biology students are like, psh, it ends in ose. Of course it's a sugar. Duh. What do you take me for, a fool? Right?
Wait, me?
Yeah.
Yeah. Wait, no. What was the question?
Just say yes.
Wait, you asked if I took you for a fool. No.
Yeah, seriously.
You guys get
And then the last thing attached up there is a nitrogen or nitrogenous base. How many choices do I have? It's like the interchangeable piece. They could be A, T, C, or G. Adenine, thymine, cytosine, and guanine. But A, T, C, and G is just less of a mouthful. Agreed? I have bad news. You're too smart for this. This is the cartoon, right? These are just shapes. My nephew's 3. He could look at that and be like, yeah, that's a circle, the pentagon, and a rectangle. He wouldn't think anything of it. You deserve to know what they really look like. So we're going to start by looking at what the nitrogen bases really look like. those are just the nitrogen base portion. So those things, I should say this in advance. No, I will never ask you to draw that on a test. Be like, yes, draw the correct structure of thymine and make sure all the double bonds occur in the correct place. That's not of a concern to me. What I do want you to know is how many rings wide they are. This takes us back to chapter 2, where I told you that chains of carbon can do crazy things, like it can be a long chain or it can fold back into a ring. Remember that discussion? That's what they've done here. They've folded into a ring shape. 2 of our nitrogen bases, and we call them nitrogen bases because they contain nitrogen. 2 of our nitrogen bases fold back to make 1 ring, and 2 of them fold back to make 2 rings. Keeping in mind every single line, every single black line you look at represents 2 shared electrons, so it's just a covalent bond. This is how I always remembered it. We classify them based on their ring size into either purine or pyrimidines. Purine is a nice short word, right? P-U-R-I-N-E. Nice and short. They have the larger structure. So they have the larger structure. Pyrimidine is a big old long complicated word. They have the nice short structure. I would have done it opposite if I were the person in charge of naming, but they didn't ask my opinion because I was like not even born yet when they named those. So I didn't get to vote. I do expect you to know that your purines have 2 rings and are adenine and guanine, and that your pyrimidines have 1 ring and are cytosine and thymine. This is going to be a really important distinction for you guys. Last hour they were like, what? They're spelled differently? Yes, there are 2 ways to spell complementary, right? And one of them means like, gosh, I really like your socks. Or, gee, you did an awesome job singing at Mass yesterday. And the other one means, well, I can add those 2 things up. They fit well. They go together. Which one's which?
The one with an I think is a compliment.
Yeah, that's the one that's like, oh, gosh, you look nice today. As opposed to this one, where things add up or go together, like a complementary angle. Complementary angles add up to what? 90?
180. 180. 90?
90.
Oh, all right.
Yep. Supplementary is 180, yeah?
Yeah.
And you thought I only knew biology. I dabble in geometry when it's convenient. That's important to you because of the way the bases are going to fit together. The bases are complementary in the add up sense, not in the, gosh, adenine, I really appreciate that double bonded oxygen you have. Not like that. Adenine and thymine are complementary. They go together always. And cytosine and guanine go together always. This is a pentagon. Which part of the nucleotide is it?
Sugar?
It's the sugar. Have we specified how many carbons that the sugar has?
1.
So organic chemists label them based on what carbon they are. So here's how we're going to label this sugar. anytime you see an angle that does not have a chemical name written on it, assume there's a carbon there. So, sound effect. See, I warn you now. Except apparently it wasn't a sufficient warning. There's a carbon right here, a carbon here, there's a carbon here, there's one here, and there's one there. You see an angle with no chemical symbol, assume it's carbon. So organic chemists are going to label the carbons like this. Where the nitrogen base attaches, that's carbon number 1. Now if we move counterclockwise, we encounter oxygen. That's not a carbon, so we shouldn't go that way. We'll go clockwise and encounter carbon number 2. Then there's carbon 3, 4, and carbon number 5. All organic chemists do this because it helps us talk about the spatial arrangement. Because remember, this happens in 3 dimensions. So they do this so we can talk about the spatial arrangement of these molecules. So we call this side of the nucleotide, because it's closest to the 5th carbon, we call it the 5 prime. That's what that apostrophe means. Just like in math when we read it as prime, we read it that way here. The 5 prime end of our molecule. If I were to look at one facing the opposite direction, 1, 2, 3, 4, 5, that's the 3 prime end. All this really tells us is which direction the molecule is oriented, up or down. Need proof? Check out where the oxygen is. In one, oxygen's pointing up. And in the other, oxygen's pointing down. Do you see that?
How do you know which one's prime? Like, this one is 5, this one is 3.
How do you know which one, like,
What is the prime?
it's just what we call it. Prime doesn't necessarily mean anything in the same way it does in math. It's just the name.
Why does it matter if one's upside down?
Because it turns out an enzyme we're going to encounter later in the chapter can only move in one direction. It can only go from 3 to 5 prime.